Hello guys, today we will talk about second lecture, cell injury. In this lecture we will be focused on definition of cell injury, etiologic factors of cell injury, pathogenesis of cell injury, and the manifestation of cell injury. Let's remember what does a cell look like. A cell is a morphofunctional unit of an organism. The cell consists of ribosome, vacuoles, mitochondria, smooth endoplasmic reticulum, lysosome, plasma membrane, Golgi apparatus, cytoplasm, nucleus, rough endoplasmic reticulum, centrioles, and microvilli. This topic is very important for us because cell damage is at the heart of every disease. Cell injury, such changes in the structure, metabolism, physical and chemical properties and the functions of the cell, which lead to disruption of life. Cells actively control the composition of their immediate environment and intracellular surrounding within a narrow range of physiological parameters. Hemostasis. Under physiological stresses or pathological stimuli injury, cells can undergo adaptation to achieve a new steady state that would be compatible with their viability in the new environment. Cellular injury may be a reversible process, in which case the cells can recover their normal function, or it may be irreversible and lead to cell death and necrosis. If the injury is too severe, irreversible injury, the affected cells die. What can damage the cell? Cell injury is the functional and morphofunctional effects of the variety of stresses on the cell from various etiological agents, which result in changes in its internal and external environment. Cellular response to stress depends upon the type of cell and tissues involved and the type and dose of injurious agent. When exposed to mild stress or due to altered functional demand, the cells adjust and adapt to a limit cellular adaptation and revert back to normal. Mild to moderate stress for shorter duration may cause reversible cell injury. Severe and persistent stress causes cell death. Among various etiologic factors, hypoxia ischemia is most important. Others, physical, chemical, and biological factors. Physical, it is mechanical, thermal effects, changes in osmotic pressure in cells, excess free radicals. Chemical factors, acid and alkalis, salts of heavy metals, cytotoxic substances and medicines, poisons. Biologicals, microorganisms, cytotoxic immunoglobulins, deficiency or excess of biologically active substance. Immunologic causes, hypersensitivity reactions, anaphylactic reactions, autoimmune diseases, autoimmune reactions. Agents. Next one, the hypoxia and the ischemia. Cells of the different tissues essentially required oxygen to generate energy and perform metabolic functions. Deficiency of oxygen or hypoxia result is a failure to carry out these activities by the cells. Hypoxia is the most common cause of cell injury. Hypoxia may result from following two pathways. First, the most common mechanisms of hypoxic cell injury is by reduced blood supply to the cells due to interruption, ischemia. Second one, hypoxia may also result from impaired blood supply causes other than interruption, disorders oxygen carrying RBCs, anemia, heart diseases, lung diseases, and increased demand of tissues. The main factors and mechanisms: hypoxic cell injury, we can see here penetration radiation, it is physical factors, inflammation, it is biological factors, chemical substance, chemical factors, hyperoxia, we can see, and the tissue reperfusion. Tissue reperfusion leads to the decreased oxygen concentration, and we can say reactive oxygen species, and finally cell damage. Here we can see cell membrane injury. Hypoxia is lack of oxygen in cells and tissues that generally results from ischemia. During periods of hypoxia, aerobic metabolism of the cells begins to fail. This loss of aerobic metabolism leads to dramatic decreases in energy production ATP. Within the cells, hypoxic cells begin to swell as energy-driven processes such as ATP-driven ion pumps begin to fail. The pH of the extracellular environment begins to decrease as waste products such as lactic acid, a product of anaerobic metabolism, begin to accumulate. The cellular injury processes may be reversible if oxygen is quickly restored, or irreversible and lead to cell death. Certain tissues, such as the brain, are particularly sensitive to hypoxic injury. Death of brain tissues can occur only 4 to 6 minutes after hypoxia begins. The loss of ionic balance in hypoxic cells can also lead to accumulation of intracellular calcium, which is normally closely regulated within cells. There are a number of calcium-dependent protease enzymes present within cells that becomes activated in the presence of excess calcium and begin to digest important cellular constituents. Sections of cell damage. Pathological agents lead to the personally damaging the cells. Next one, it is individual subcellular structures and components of the cell and intracellular interactions. General mechanisms of cell damage or classification of the mechanisms. It is energy disruption, damage membrane and enzymes. Next one, imbalance ions and liquids, cell genetic disorders, and disorder mechanisms regulation. First, general mechanisms of energy disruption. Actions of pathogenic factors, biological, chemical, physical, different other factors lead to the reducing the intensity of ATP in resynthesis, glycolysis, and tissue respiration processes. Disorder of ATP energy transportation mechanisms from the places of its resynthesis to the structures of the cell. Impaired use of energy ATP effective structures of the cell. All this leads to the disorder of the cell life. Second, general mechanisms of cell membrane damage. Action of pathogenic factors leads intensification, free radical reactions, and free radical lipid peroxidation. Activation of lysosomal membrane band and hydroxylase. This together leads to the accumulation of excess of products, free radical lipid peroxidase, hydrolysis of lipids and phospholipids, which leads effects of excess food, free radical lipid peroxidase, hydrolysis of lipids and phospholipids. Impairment of conformation of protein, lipoprotein, phospholipid molecules. Inhibiting resynthesis processes of damaged membrane components and or synthesis them again. Stage and ruptures of membranes of a hyperhydrate cell. all this leads modification of physical chemical properties and biochemical composition of cell membranes, violation of their structure, and again finally disorder of the cell life. Imbalance of ions and liquids in a cell, their damage. Action of pathogenic factors leads output potassium and magnesium from the cell and accumulation sodium, hydrogen and calcium in the cell, which leads to cell hyperhydration, and this pathogenic factor leads cell hypohydration. Together they leads transmembrane ion imbalance and disruption of the ratio of individual ions in the hyaloplasm and intracellular fluid. Mechanisms of calcium-dependent cell damage. Damaging agents, physical, chemical, biological, leads to the increasing calcium in the cytosol. It leads to some mechanisms. First one, it decreases ATPase, which leads to the ATP deficiency. Next one, increase phospholipase, which leads to the destruction of phospholipids. Next one, increase proteases, which leads protein degradation. And the last one, increase endonuclease, which leads to DNA fragmentation. Mechanisms of cell genetic disorders. Action of pathogenic factors leads mutation cells, expression of the pathogenic gene, disorder of expression of the normal gene, disruption of processes of transcription and of translation of genetic information, disorder of nuclear and cytotomic processes, implementation of a fragment of alien DNA. Here we can see three cells. First, this is a normal cell. This is a cell after reversible damage. This is after irreversible damage. They are different. In normal cell, we can see normal Zeiss lysosome, Zeiss and form lysosome, nucleus, and plasmic reticulum, mitochondria. Reversible after damaging, this cell is changed. Form is different. There is very big, and you can see a protrusion of plasma lemma, cell swelling, chromatin fragmentation in the nucleus, lysis of organelles, mitochondria by the lysosomes. Here we can see membrane damage after damaging etiologic factors, swelling of the endoplasmic reticulums here, and the separation and dispersion of ribosomes from the endoplasmic reticulum here, and the swelling of mitochondria and the calcium deposits there. Irreversible damage. Rupture of the lysosome membrane and autolysis. If here just we can see protrusion and deformation, here we can see autolysis, pycnosis and of the nucleus, lysis, destruction of it. Dystrophy. In reversible we don't have dystrophy. In irreversible we have dystrophy. Endoplasmic reticulum lysis. Membrane defects also, here we can see just membrane damage. Here in irreversible it is membrane defects. Increase in deposits, swelling of mitochondria. Cell death, cell injury leads to the reversible injury, cell recovery and return to normal function. Apoptosis and programmed cell removal, cell death and necrosis. Like disease, cell death may be caused by internal intrinsic factors that limit the cell's lifespan or external extrinsic factors that contribute to cell damage and aging. When a stressor is severe or prolonged, the cell can no longer adapt and it dies. Cells death or necrosis may manifestate in different ways depending on the tissues or organs involved. Manifestation of cell damage. It is dystrophy, dysplasia, disorder of subcellular structures, necrosis, and apoptosis. What is a dystrophy? This is a metabolic disorder. They are accompanied by a disorder in the functions of cells, plastic processes in them, and structural changes lead to the disruption of cell activity. Dysplasia. Disorders of the cell differentiation. They are accompanied by persistent changes in the structure, metabolism, and function lead to the disruption of the cell activity. Types of cell death and the mechanisms of their destruction. Action of pathogenic factors. We can see action, physical, chemical, and biological different factors leads to the cell necrosis or cell apoptosis. If it is cell necrosis, it leads to the lysis of a necrotic cell, necrolysis. If it is cell apoptosis, phagocytosis of the apoptosed cell. Apoptosis. The form of death of individual cells arises under the influence of extracellular or intracellular factors. It is carried out by activating specialized intracellular processes. It is regulated by certain genes. In Greek word apoptosis, falling leaves. Apoptosis is genetically programmed cell death. Common event in some regenerating tissues such as skin and gut epithelium and during embryogenesis. Not a typical event in developed tissues such as brain. Becomes a serious occurrence when many cells are involved in such organs as the liver. Example of the pathological apoptosis. It is irradiate tissues, viral infections, viral hepatitis, hormone-dependent atrophy of tissue, endometrial cell breakdown after withdrawal of estrogen and progesterone in the menstrual cycle, death of tumor cells by cytotoxic T cells. Stages of apoptosis. We have four big stages: initiation stage, programming stage, realization of the program stage, and removing dead cell stage. Apoptosis. First stage is initiation stage. Pathogenic factors, extracellular and intracellular. Extracellular, it is negative, deficit growth factors, enzymes and hormones. Positive, tumor necrosis factor, mixed, nitrogen, antigen, or others. Intracellular physical chemical agents, acidosis, excess free radicals, increased temperature, viruses, hepatotropic, neurotropic, hormones penetrating into the cell, steroid and thyroid hormones. It leads to the second stages, apoptosis programming stages. Programming stages, realization of the effects of the causes of apoptosis, direct and indirect. Direct adapter proteins, enzymes, cytochrome C, and indirect cell genome, gene expression of apoptosis, repression of the anti-apoptosis genes. Gene expression of apoptosis leads proteins promoters of the apoptosis. Repression of anti-apoptosis genes leads proteins inhibition of apoptosis. This is all leads to the affected caspase endonucleases. And starting next stages. Implementation of the apoptosis program. Stages of program implementation: implementation of the apoptosis program, activation of effective case spaces, and endonuclease activation. Activation of effective case spaces leads to destruction of cytoskeleton proteins, destruction of nuclear structural proteins, destruction of regulatory proteins of the nucleus. Endonuclease activation leads DNA fragmentation. All this leads to the destruction of the cell, formation of apoptose bodies, and the next stages phagocytosis of dead cell fragments. This is a normal cell. This is a damaged cell. And here we can see difference between necrosis and apoptosis. Phagocyte, here we can see phagocyte. This is the apoptotic bodies. Manifestation of cell damage. Action of pathogenic factors leads damage to the cell. This is a least manifestation of the cell damage. Specific and non-specific. Specific for a certain cell, a certain factor. Non-specific, specific for various factors damaging them, various types of damaged cells. Types of necrosis: liquefactive necrosis in brain, caseous necrosis, infection with Mycobacterium tuberculosis, fat necrosis, liver and pancreas, coagulative necrosis of kidney, liver or heart muscle, and gangrene necrosis of an appendage, usually limbs. Pathology of necrosis or necrotic chains. When a cell dies, enzymes inside the cell are released and start to dissolve cellular components. This triggers an acute inflammatory reactions in which white blood cells migrate to the necrotic area and begin to digest the dead cells. Now little bit I want to tell about enzyme markers of the cell death. From a laboratory we can check these enzymes. It is aspartate aminotransferase, alanine aminotransferase, and creatine kinase, amylase, and lipase. They are marked the cell death. This literature used for presentation is Litvitsky Pathophysiology. You can use this literature book for practical your classes also. Robbins and Katran, Hashmohan textbook of the pathology. Thank you so much for attention, guys. Bye, see you.